Hello and welcome to another episode of Momento Malum today. At present, the chicken is a key species in the human food system, as well as a common model organism for research. As a result of different domestication events and different breeding projects, divergent breeds of chicken from heirloom to commercial exist in the current gene pool. There's a controversial debate on whether the domesticated chicken is a descendant of a single ancestor population of red jingle fowl in Southeast Asia, or if it was domesticated multiple times. Local chickens native to various countries in Africa, and local middle-producing breeds represent a diverse and unique adapting gene pool. The standard fancy breeds were utilized to synthesize the cross types that were utilized in experimental inbred and highly specialized commercial chicken breeds, such as the egg layers and broilers. The first chickens brought into Europe were the Mediterranean types that were improved over time in ancient Rome. Following this, Chinese and Malaysian breeds were introduced. Much later, with the massive use of selection and crossbreeding, local breeds in different lines were bred in Europe and further developed. Many of these European breeds were introduced to America via the Spanish in the 15th century, and much of our breeds are derived from these European lines. In the modern era, Asia and Europe have more than 400 local chicken breeds. Africa and Latin America report more than 100 each, and the Near and Middle East and North America report fewer than 40 each. The extensive breeding efforts of chickens due to commercialization, in combination with new diseases that have been popping up, have brought various chicken breeds under the threat of extinction. As a result, Dr. Eltanany would gather all the available information regarding the gene pool of chickens to determine the genetic variation within the domesticated chickens, and here are the results. The chicken was the first domesticated animal to have its genome sequenced. Earlier studies have revealed that the bird genome is approximately 1/3 the size of a mammal genome. It was found that 70% of the genetic material across chicken breeds are found in all breeds. This suggested that the domesticated population of chickens was closely related prior to domestication, around 5,000 to 10,000 years prior to the full domestication process. The second possibility was that their ancestry is no longer knowable due to extensive breeding between Asian and European lines. Using the archaeological records, the chicken origin is likely the red jingle fowl that was domesticated as early as 5400 BCE. In addition, the records of cockfighting behavior in ancient pictorial assembling, supported by various historical documents, found that this species was likely domesticated for the same reason dogs were: ornamentation, companionship, and sport. One of the first attempts to look at the genealogical origin of the present domesticated populations happened in 1994. The result of the study was the single origin model of domesticated chicken. This model suggests that a red jingle fowl subspecies in a region of Southeast Asia was selectively bred to breed chickens. This result was further supported by an analysis using the SNP genetic marker technology. Based on this information, the domestication event of chickens happened in a Thai population of red jingle fowl. In 2005, a competing model known as the polyphyletic model was formed based on the sequencing of the entire mitochondria of the chicken breeds, as well as a genetic analysis of other species of jingle fowl. They found ancient hybridizations between the gray jingle fowl and the red jingle fowl, and between the gray jingle fowl and the silent jingle fowl. This created the idea that chicken was domesticated from a population of red jingle fowl with minor traces of gray jingle fowl DNA. A later study found that the yellow skin of many chicken breeds does not originate from the red jingle fowl, and is more likely a grey jingle fowl trait. Moreover, the Sri Lankan indigenous chicken had higher genetic similarity to the red and grey jingle fowl than the silent jingle fowl. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. Using different genetic marker technologies, one study found that a chicken gene pool could be split into 6 different population clusters. The Asian and European breeds represent 2 clusters. The cluster consisting of the commercial broilers and brown egg layers were of mixed ancestry, containing both Asian and European pedigree. The white egg layers made up the 4th cluster, and was wholly derived from a narrow population of European breeds. The remaining 2 clusters had no overlapping pedigree of the other groups. A further study regarding the breeds from the Near East and Europe found that the Northern and Western European breeds of chicken were of a different lineage to the Near Eastern, Eastern European, and Southern European breeds. The genetic difference between these 2 groups was 34%. It was also found that a great many breeds have maintained a large percentage of their genetic diversity over time, and the breeds from Hungary, China, and France are the most genetically diverse populations, with France having the largest gene pool of chickens overall. Using SNP marker technology, Dr. Andrescu found that the commercial lines have only half of the genetic diversity found in non-commercial breeds. Another study in Africa found that across multiple countries, there's an extensive level of disease resistance on a genetic level within their breeds, as well as a larger than expected size gene pool. Overall, Dr. Eltanany found that the breeds from China, Southeast Asia, France, and Africa had the widest gene pool, and the commercial breeds had the least. It was also found that overall, the chicken gene pool is still quite wide, and we can be grateful for that. And that about covers everything. Thank you for watching.